of couple oscillators for the parcellation of the brain's pial network. Please.
Hello everyone, so I am Marie. I'm a PhD student, as you said, at Laboratoire de l'École Normale Supérieure de Physique in Paris. And today, as you might be surprised by, I'm going to talk to you about coupled oscillators. So first, maybe I can, yes, okay, like this. I can maybe show you who I work with. So this is Massimo Vergasola, he's my supervisor. Here is a collaborator called Massimo Cencini. He works in Rome. He works with us on the theory for this project. And then you have here the team of the laboratory of Dr. Kleinfeld, which is in San Diego. And they do all of the experimental work, which I don't do at all. So maybe first to maybe convince you that this is an interesting topic and project, I would like to introduce you with why kind of we are doing this and collaborating with Dr. Kleinfeld. So as you've been told maybe this morning, you know that when you do an fMRI, you have these kind of bright spots and you're like, oh, there is neural activity here and here, and oh, nice. But what you're doing is not actually measuring neural activity directly. You're seeing an increase in blood flow, and the fact that you can say, okay, it means that there is neural activity here. It is because of this phenomenon called neurovascular coupling, which is that, okay, you locally have an increase in neural activity, and then blood flow increases, and la la la. So that's nice. And maybe if we wanted to understand a bit better these fMRI images, something that would be nice is to understand better this neurovascular coupling, no? So what we've been working on, like the actual system that we are focusing on, is this pial network. It's called the pial network. It's a network of arteries that's at the top of your brain. Here is the brain of a mouse, hence the weird shape, you know, maybe you already know. And these pial arteries, they're okay at the top, and then they branch into the brain, into smaller and smaller vessels, eventually bringing blood to the whole cortex. And these pial arteries, they display vasomotion, which is an oscillation in their diameter that is intrinsic. Meaning that if you cut it, you take it out of the brain, you look at it, it will be doing this oscillation. But this oscillation is not only intrinsic, doing its stuff without communicating with the rest of the brain, it's also driven by a certain frequency component of the neural activity, which is I will find the word, the power of the gamma band, frequency, okay? But I will say neural activity because it's easier. So it is entrained, okay? It's work done by Kleinfeld, the laboratory of Dr. Kleinfeld. You have this entrainment between the two. Okay, and as you know, maybe, neural activity is not homogeneous in the brain. Otherwise, you're kind of in trouble, I guess. I don't know. Which means that maybe this frequency is not the same everywhere. And since you have this locking that is local, with the oscillation of the blood vessels, with the vasomotion, you end up with different frequencies of vasomotion in different regions of the network, no? Okay. So maybe we would like to try to understand that and understand why is it like that, maybe trying to be able to go from one to the other, and maybe trying to understand what happens at the boundary, something like that. So to do that, we would like to make maybe some model that would, okay, take into account all of this. So for the model, you have this pial network that you would say are oscillators that are like self-sustained. They do their oscillations by themselves. With some coupling, they talk to each other. But also, there is some either forcing or parametric variation, meaning that like this is for the neural activity, like what the neural activity will do. Either you take it as a forcing, or you take it as something that comes and changes the parameters of the pial oscillators, no? So this is how you could think about it. And what we did actually is to say, okay, it comes and maybe tunes the parameters of the oscillators, but okay, it's quite similar to do the two things. Maybe going to another topic, it's not the only system in which you have parcellation and different frequencies, different frequency clusters. Actually, there is this old paper about the intestine, where what they do is you take the intestine of a cat, dog, monkey, super nice, and you measure, yeah, I know, and you measure the electrical activity of the muscles that surround it. And you know, this electrical activity, it oscillates like this because these muscles, they contract. And this oscillation is not the same everywhere. It depends, you actually get clusters of different frequencies. So maybe here you have a certain frequency, and then a bit further you have another frequency, and in the middle you have something in between. And actually in the middle you have these weird waxing, they call it waxing and waning patterns, okay? That actually are not explained right now theoretically in the literature. They can reproduce these clusters, but not the waxing and waning. And the model that we talked to you about is actually relevant for this. They are two very different systems, but I guess you can understand that we can model things for different systems with the same kind of equations, which is pretty cool. Okay, so let's go back to this brain and pial network. Okay, so maybe we could have this continuous oscillatory system, meaning that you would, I don't know, this square, where at each point you have a complex amplitude that is defined. Complex amplitude meaning you have this amplitude times exponential of I times the phase. And then you could say, okay, maybe here the natural frequency is F1, and maybe there it's F2. But that's already complicated, and we are physicists, so we first go one-dimensional. Not just because it's also complicated, but also because it's relevant for this intestine system that I just talked to you about. So we go one-dimensional, and we have our continuous line of oscillators, where at each point you have an amplitude that is defined, a complex amplitude. And maybe here the natural frequency is F1, and maybe here the natural frequency is F2. But one question could be, okay, probably super far away, the oscillator will be doing their oscillation at F1 and here at F2, but what happens in the middle is maybe less trivial, maybe less clear. We have to figure this out. And actually, I told you about this a bit before. You have this waxing and waning paper that has not been really captured by models until now in the literature, so what is it that happens? So this is the model, and what we do is we make something a little bit like in the intestine. You have the continuous line of oscillators, and then the natural frequencies, we take them to evolve linearly in space to maybe study what happens, I told you, in between those regions. And then to account for the locked clusters on the sides, you take fixed boundary conditions here and here. So the system has finite size. And the equation, it's okay if you don't understand it, and if you do, it's cool. It's the real Landau-Ginzburg equation. Real in the sense that the parameters are real, not the A, which is a complex amplitude, as I told you before. So if you remove this term, it's the equation, but then we added this gradient in natural frequencies. And the coupling here, we chose it to be diffusive, but of course, maybe it's not. We still have to, check. And then we take these fixed boundary conditions that I told you about. Do you know how much time I have left? Do I know? Okay, thank you. So we take the equation, and we're not going to start doing computations right away, so we put it in the computer and look at what happens. And what we did, for example, is to, look at each point, what is the actual oscillation frequency, because each point has a natural frequency, okay, but maybe it doesn't actually oscillate at its natural frequency, and indeed, you get clusters, or plateaus, as we call them, a lot, of frequency. So it will depend on the coupling, the number of plateaus, as you can see here. It will depend also on the other parameters. It's a bit the same to change the length, etc. Well, I will not go too much into details, but maybe for you to understand, you can see that the limit cases are the one where the coupling is 0, so each oscillator is independent from its neighbors, so each oscillator will oscillate at its natural frequency. And the other limit case is super big coupling, so they all want to be coupled, but the boundary condition will not allow this, because you have fixed oscillation at a certain frequency here and another one here. So it has to have two frequencies, and due to the symmetries of the system, you end up with these two plateaus. For other boundary conditions, of course, you can have full locking. And then you have all of the cases in the middle, you know, another number of plateaus is possible. Okay, so that's for the number of plateaus. And then what happens in between plateaus is interesting, as I told you, for this intestine system, because it's not super trivial. It's not like the oscillations are the same everywhere. Actually, in between plateaus, you get what are called space-time defects. So I will explain to you. This is, at this point here, the blue line is the real signal, something that you would measure, you know, if you were doing the experiment. Then in red, it's the amplitude of the signal, and in orange, it's the phase. And what you can see is that regularly, the amplitude goes to 0, and at the same time, the phase jumps. These are the space-time defects. And why do they appear? Well, this is the simple case with two plateaus. And you can see that here, at this point, you will have oscillation at GL/2 frequency, and here minus GL/2 frequency. But here, you know, this system is continuous. So here there is a phase gradient that is defined, and this phase gradient, it grows and grows and grows in time if you just have these oscillations that go on forever. But that's a problem, because if you look a bit at the equation, this is the one I showed you before, and you just write A equal A times small, big A equals small a times e to the I phi. You get two equations, and you have this term here, where there is the gradient of the phase. And this one, you cannot let it go to infinity, diverge like that. No, the system will not allow it. So it will put A to 0, the amplitude. As I told you, it goes to 0, so that the phase is not defined anymore, and it can jump. And then it resets the phase gradient. And it does it regularly. So it always happens when you have the plateaus, always. And it is something that is not captured by phase models, of course, because you don't have an amplitude. So all the phase models that were used previously, maybe in the literature to look at these intestine clusters, they will not get these defects. Okay, that actually, you can see maybe are here, you know. At least in our model, it corresponds to that sort of behavior, this waxing and waning. And so we tried to match the two. So this is the data from the paper. You know, you have the plateaus, and you can have these oscillations. They would look like this. And if we try to make them match with our plateaus, so this is the line of the simulations, you can see that you can get things that sort of match. It's not a fit as in a linear fit where you can easily get the perfect parameters. There are so many parameters possible, so maybe this is not the best fit, but you know, it works, and there are many that that were kind of working, and you also get this waxing and waning thing where you have the defects, so between the plateaus here. So that's pretty cool. Maybe I don't have time to talk about everything, so before I go to maybe a little bit more, I have, 1 minute? I can tell you what you expect to find in the paper that we just submitted, so it's not yet online, but if we are interested, you can maybe look it up one day. We study the number of plateaus and their evolution. We study how the defects appear and get some results about this. We look at the stability of the A=0 state for other boundary conditions, the no-flux ones. And we capture amplitude and phase in special regimes, like we can actually calculate it. And then we understand how the nonlinearity affects the full locking regime. Okay. I will not explain everything. Like, this is kind of the picture for the number of plateaus for different parameters. So you see that it's not completely crazy. There is some logic in the evolution. Well, anyways, I will not bother you with this. Okay, thanks a lot. And that's the name of the paper if it goes out one day, I hope. Thank you.
Okay, thank you very much for the talk. Mario.
Thanks. Thanks, Marie. Very, very interesting work. A very nice presentation. Thank you. I have very few curiosities, and the first one is related to probably to the first or your second slide, because you put from the, for you, you assume that is the neural activity who drives the vascular coupling. But I'm wondering if you have considered probably the effect, this one,
Yes.
if you have considered probably the effect of the cardiac dynamics.
So yes, I think what they see, but maybe I could show you or check, is that you have a bit of like this vasomotion. If you look at the spectrum, there is the cardiac dynamics, but it's not the same frequencies, and you can, you can discriminate between these cardiac effects and also the ones of this frequency in the neuronal activity, because this is around 0.1 hertz, so it's very slow.
Thanks. And my second curiosity, sorry, Albert, is about what you observed from the data and from your modelization of the waning and waxing dynamics. You can observe a kind of modulation. And do you have an idea of to what corresponds the slow modulation? No, this is something that is here, a kind of bumps. This frequency of the bumps, do you have an idea?
The frequency of the, so it's like the frequency of the defects. And
but physiologically, does it correspond to something for the?
Yeah, so every time there's a 2 pi difference between two plateaus, the phase gradient has to be reset. So the frequency of these like jumps will be, I would have to know again, like GL over 2 pi for the whole system, but that depends on the exact difference between the two plateaus that you have, if they are clean, because sometimes the defects, they move. Well, anyways, I can explain also later if you want.
Okay, any other question? Okay, in this case, let's thank again the speaker. Thank you, Marie. Probably now we move to the coffee break, Hilda. Move to the coffee break.